Okay, so for this part of the course, what I'm going to focus on is this idea of efficient gas exchange systems. How can we have a system which maximizes the amount of gas moving from one direction to another? So, when we're talking about gas exchange, we're really focusing mainly on the movement of oxygen and the movement of CO2. So things we have to be able to appreciate is what adaptations exist that help to maximize and make the system most efficient. So one of those areas is having an increased surface area, and the example you need to know is the root hair cell. And clearly this cell here, if that cell was just like that, you could see it would have that surface area. But by having this hair protruding out, it's increasing the surface area significantly, and that's going to be important not just for gases like oxygen to get in, because root hair cells, they need to be able to respire as well, so they're going to need a supply of oxygen. This is also going to be helpful, and later on you'll learn about this, the absorption of water as well. This is going to help to maximize the amount of water that gets into the roots. So that's going to help to get gases moving. If you look at this alveolus here, this shape here, this spherical shape is helping to increase surface area. And just looking at the lungs, you've got millions of alveoli within the lungs. So all these little tiny little spherical shapes, all filled with air, they're all working together to significantly increase the surface area of the lungs. We're talking about a tennis court's worth of surface area because of their combined shapes working together. And then if you look at, you have to be aware of increased surface area in fish as well. Well, they've got gills, and gills, you can kind of think of them as a bit like villi and microvilli in the small intestine. So you've got this gill arch with these filament-like structures. Each of these individual protrusions are helping to increase surface area, and each one of these little filaments, little lamellae parts here, they've each got their own blood supply, just like a villus would as well. So it's maximizing the amount of gas that can get into the bloodstream. And then if you focus on each of these little filaments, you can actually see there's even smaller structures called the secondary lamellae. All of these things all working together to increase surface area. And look at all the structure of this gill here, all those lines, they're all giving you a sense of how this is much more effective than just one smooth sheet where gas exchange would occur. A thin layer is also going to be really important. So if you look at diffusion is the movement of particles from a high concentration to a low concentration. Well, one way to make that journey as easy as possible is to make the boundary as thin as possible. So the alveoli, that sac structure there, it's only got one layer of cells acting as the boundary. So it's only one cell thick, these epithelial cells. So this is going to make the journey which oxygen has to make to diffuse into the bloodstream much shorter. And same for the CO2. You need CO2 to be able to diffuse out as well in order for that to happen. So let's see what else as well. So increased surface area, this root hair shape here. We've got the large number of alveoli in the lungs, and we've got the gill filaments in the fish. Thin layer, one cell thick of the epithelial cells. Okay, good blood supply is also going to be really important for maintaining a concentration gradients. So in order for diffusion to occur, this is only going to continue happening if there is a diffusion gradient, there has to always be a high concentration and a low concentration. And if that difference in concentration, if it becomes equal, then diffusion stops. So you've got a situation here where if we look at this, the oxygen coming into the blood here now, the reason why oxygen is moving across the epithelial layer into the blood capillary, which is running next to every alveoli, the only reason why it's continuing to move this way is because of the concentration gradient, which means the region where the blood is, there has to be a low oxygen concentration, and in the alveolus, there always has to be a high. So why is there always a low concentration of oxygen in the blood? Why is it that if oxygen is diffusing in, surely that equilibrium should occur and there should be no more diffusion? Well, these individual blood cells here, they are going really high up in concentration, but luckily, because of the heart pumping, these blood cells are being moved away and they leave. They're gone. So any chance of an equilibrium occurring isn't going to occur now because they're gone. They've been pushed out of the way. And what's replacing them? Blood cells, which are deoxygenated, have a very low concentration gradient, and therefore you're going to keep maintaining this low. It's always low here. As long as the heart keeps pumping, there's always going to be a low concentration of oxygen in the blood when it arrives at the alveoli. So why is there always a high concentration of oxygen in the alveolus? Well, that's because we're constantly breathing. We are inspiring oxygenated air, so it's oxygen-rich air is entering the alveolus. That oxygen is diffusing, so the concentration is going down. So this level of high is getting less high. It's also starting to gain CO2. And then what do we do next? We breathe out. We get rid of all the air which is low in oxygen. And what do we bring back in? We bring back air which is high in oxygen. And that's going to maintain this high concentration. So breathing is maintaining a high concentration of oxygen in the alveoli. And the movement of blood, due to the heart pumping, is maintaining a low concentration of oxygen in the blood capillaries, and therefore we're always going to have this movement occurring. This is a really good thing. This is taking place in order to achieve a high concentration gradient. And the same happens within fish as well. So fish, they need to get their oxygen as well, and there's oxygen dissolved in the water. And you can see here, when the fish is in the water, it takes water into its mouth, it opens it really wide. Now, you've seen fish open and close their mouth. And when they close their mouth, they force water out the side of their face. They've got these flaps here called the operculum, and water is being forced out the side of their face or side of their head, and that's exactly where the gills are. So water is forced over these gills. And what do these gills have? Well, they're red, aren't they? So they're really rich in blood. So the fish is sending blood to the gills, just like we send blood to our lungs. And when that blood arrives, it's going to be very low in oxygen because it's been used by all the cells in the fish for respiration. So here's our blood, which is low in oxygen, is entering the gills. The water is flowing across the gills in one direction, and then at the same time the blood is flowing the opposite. We call this the counter current flow. So blood is going the opposite way to the water. So if you think about, this is really important, this is a really good way to maintain a concentration gradient. If you imagine blood moving along the length of these gill filaments, if oxygen were to be moving in the same direction as the blood, well, this would be a problem, because imagine they both arrive, the water containing the oxygen will be high in oxygen, high in O2, and the blood is low in O2. So initially it's good, because you can see here, as they arrive, the oxygen will diffuse towards the blood in the gills, but very quickly equilibrium is going to occur. So the rest of this journey of the blood is wasted. It can't get any more oxygen. So now for the rest of this blood's journey, it's just in the way. It's pointless it being there. So rather than having a system going this way, if we imagine the water's going this direction, the blood moves the other direction. And what's useful about this then is that this is low, and here it's high. When the water, which is high in oxygen, arrives, it can still give oxygen to the blood, because it's got such a high concentration here. In relation to what's happening here, it's still higher. And as it starts getting lower and lower and lower and lower, it can still give oxygen, because the blood is so low in oxygen that anywhere along the length of this filaments, the blood always has a lower concentration of oxygen than the water does. So this counter current flow is helping to maintain a concentration gradient. So fish are doing it with their counter current flow. We're doing it with our blood constantly moving. It's never allowed to stay still. This is helping to maintain a concentration gradient as well. So last little thing to mention as well, in order to maximize the efficiency of gas exchange, or maximize the efficiency of diffusion within the gills and the lungs, there's a really rich blood supply. So any oxygen that does get into our lungs or into the crop that passes over the gills of a fish, there's much more blood now for it to be able to diffuse into.